Okay. Yeah.
Good morning, everybody. I hope you can all hear me. You're in the session where we are going to hear two speakers. The session 7, that is Sectional Committee 7. And it's my pleasure to introduce Professor Raghavan Vardarajan, who did his PhD from Stanford University and his postdoc at Yale, where he worked with Professor Richards and became an expert on the thermodynamics of protein folding and cavity formation. So he's taken that experience and interest to great heights in his own career. He joined IISc in 1992, spent his entire career and continues to be a senior professor at IISc. He also has done a lot of work for IISc in administration, chaired the Department of Molecular Biophysics, and has been honored for his research, many honors. He is a fellow of all the academies. He's a Bhatnagar Prize awardee. He's held the J.C. Bose Fellowship, got the B.M. Birla Prize, and when he was a lot younger, he also held the Swarna Jayanti Fellowship and Wellcome Trust International Senior Research Fellow. In today's talk, he's going to be very contemporary in his seminar. He's going to talk to us about how he's taken the design principles of protein fold and put it towards developing a subunit vaccine. Raghavan?
Okay, thank you so much, Usha, and thanks for the introduction as well as the opportunity to talk over here and talk a little bit about some of our work. Now, I'll give a brief sort of motivation and background to the rest of the work. So basically, we study, I don't know how general the audience is, but in any case, we study proteins, which are biopolymers composed of 20 different amino acids. They vary in length and they have a specific amino acid sequence, which is dictated by the gene sequence. And that amino acid sequence controls their three-dimensional shape. And the sequence contains sufficient information to specify the shape. And this is one of the versions of what is widely known as the protein folding problem. And recently, just within the past 6 months or so, or over a year, there have been real advances in using machine learning for protein structure prediction, that is going from the amino acid sequence to the structure of the protein. However, predicting effects of point mutations on protein stability, it remains very challenging. And it's an apparently simple problem, but it is not has not really been solved in a comprehensive way. And one of the problems is that the stability of a protein is a small difference between two very large numbers, the stability of the free energy of the folded state and the free energy of the unfolded state. And so using computations, it's very difficult to predict the effects of mutations on stability. And my lab works on understanding the relation between sequence shape and stability. And we apply this knowledge to design proteins for various applications. And here I'm going to talk about our work on viral vaccines. Now, these arrows don't seem to work. Sorry about that. Okay, maybe.
Raghavan, right at the bottom.
Yeah, I see that. Yeah. So this just summarizes the main areas in the lab. And what I will do is be focusing really on our work on COVID vaccines. Okay. So now I'll give a brief overview. This is unrelated to, well, it's not, this is not the vaccine work, but it feeds into the vaccine work. So we have for a long time been interested in understanding what kind of information we can get from mutational data on proteins. So the idea is that supposing we had the phenotype or how every mutation in a protein affected its properties, such as binding or biological activity, could we predict the structure of the protein? And could we use this information to design more stable proteins? And the way this is done is we create a mutant library, and we develop methods for making large numbers of points mutants in parallel in an efficient way. And then we, once we have the library, we subject it to various kinds of assays. And what this allows us to do, so we take this mutant library, put it into an organism, uh, typically bacteria or yeast, and then we assay the phenotype of the organism. That is either its ability to survive under certain conditions or its ability to bind another protein, the protein inside the organism, to bind some target protein. And then what we can do is we can, so therefore at every position in the protein, we know the effect of every possible mutation. So if there are 100 residues and you have 20 substitutions per residue, we can get information on close to 2,000 mutations in a single experiment. And this is encoded in the form of a heat map, where these are the residue numbers and these are the 20 different kinds of amino acids. And this measures in a very, it gives a compact representation of the effect of each mutation at each position on whatever property you're measuring in the phenotypic assay. And so this is nowadays called deep mutational scanning. We had done some early work in this area. It was first implemented on a small scale by Doug Fowler along with Stan Fields, and then we were the first to apply this to a whole protein and get this kind. Now it's a very widely used methodology. So I'll talk about another technology, which is yeast surface display. So in yeast surface display, we express a protein of interest and express it on the surface of yeast cells. And in general, this will be a library of different mutants. Now we can detect the level of expression of the protein by an antibody which binds to a tag before the main part of the protein of interest. Then we can add a binding partner, and the binding partner has another tag onto it. And then we can also detect binding of that tag with another fluorescent antibody. So what this allows us to do is for each yeast cell, we can measure the amount of displayed protein as well as its ability to bind to a binding partner. In this case, just for representation, it was a protein we work with called CCDB, and its binding partner is a protein called DNA gyrase. And then we can have a double plot where we have both these fluorescence events depicted. So this is for a single protein. We can do the same thing for the mutant library, and then we get a slightly more complicated plot. And so we can measure the mean fluorescent intensity of every yeast cell in the population, and each yeast cell will be displaying one particular mutant protein on its surface. Okay. So now, when we look at when we get this kind of data, mutations at both buried residues in the interior of the protein, as well as protein residues which are directly involved in binding, they affect binding to a binding partner. And if you don't know the structure, discriminating between these two classes of residues becomes challenging. And so we recently have developed methodology to distinguish between these two different kinds of residues. So this is what you get basically in a heat map. Blue means the binding to the binding partner is severely affected. Red means it's not affected. So again, these are different residue numbers. These are the different substitutions at each position. So all of this data you essentially get in a single experiment. And the way you interrogate it is through deep sequencing. So instead of measuring the fluorescence of every mutant, you measure the fluorescence and histogram, and then take slices through the histogram and sequence the mutants in that slice of the histogram through next generation sequencing. Okay. So, and what you can see is that for residues which are buried, both the expression as well as the binding are affected, whereas for residues in the active site, the expression is not affected at all, but the binding is affected. And so even in the absence of structure, this allows us to discriminate between these two classes of residues. And this work is recently published. Now, using these yeast screens, you can get information on binding activity, but it's difficult to isolate more stable mutants from such libraries. So we developed a methodology called second site suppressor mutagenesis. And in this kind of methodology, what we do is we start out with, we have a library which has all possible mutations. And into it, we introduce a mutation at a buried position, which leads to a loss of function. And then we can screen for suppressors, as, which so we are able to distinguish between these two classes. I don't have time to describe how, but the proximal suppressors allow us to identify residues which are in close proximity, and the distal suppressors identify stabilizing mutations. And so we applied this to CCDB, and using this suppressor mutagenesis, coupled to deep sequencing, we isolated large numbers of stabilizing mutations. So in black is shown the wild type, and using suppressor mutagenesis, we were able to isolate all these different stabilizing mutations, and some of them you can see stabilize the protein by quite a large amount. Especially when we could combine single and double mutant, multiple single mutants to get stabilized double mutants. And we could even get combine individual stabilizing mutants. And finally, from when we had 6 mutants put together, we got a stabilization of about 20 degrees or so. And for up to 3 mutants, we got a stabilization of 15 degrees. So we can, this is just to show we can use this methodology to very rapidly isolate stabilizing mutants from these libraries in the absence of any structural information. So now I'll turn to the topic of COVID immunogen design. So again, a very, very brief primer on immune responses to infection. When an infectious organism, such as a virus or bacterium or other pathogen enters the body, the first barrier it encounters is innate immunity, which are physical barriers, as well as certain kinds of cells in the immune system, which recognize general patterns on pathogens. So what are called pathogen-specific patterns. If the infectious organism is able to escape innate immunity, then the adaptive immunity comes into place, and B lymphocytes or B cells recognize the intact antigen, and ultimately you get antibodies which bind to proteins on the pathogen surface and prevent it from infecting cells. Okay? So this is the humoral or antibody-mediated arm of the immune system. Of course, some copies of the pathogen will indeed infect cells most of the time. And what the cellular branch of the immune system does, apart from interacting with the humoral branch, is to recognize infected cells which display small fragments of the pathogen on their surface and target those infected cells for destruction. So in the case of coronavirus, the major protein on the surface is the spike protein, which gives it so-called, corona name. And what the spike protein does is it binds to the ACE2 receptor on the surface of target cells. And this initiates the process of viral infection. And one of the first steps is the fusion of the viral and the host membranes. So if antibodies can prevent this initial receptor binding step, right, then that will abort the process of infection right at the beginning. And most vaccines are focused on eliciting protective and preferably neutralizing antibodies which target the spike glycoprotein. Okay, so this is a very brief overview of the situation with the different kinds of vaccines. There are multiple, there are about 14 to 15 vaccines in use in different geographies. These include 2 mRNA vaccines for viral vector vaccines, inactivated virus, and 1 recombinant protein vaccine. So this is the situation in India. We have about roughly 35% of adults who are fully vaccinated, and worldwide the number is slightly higher. And of course, sero surveys estimate a considerably larger fraction of the population has been infected. So now, if you look at both the phase, the clinical trial data, as well as evidence of real-world efficacy, so what we find is, just to summarize across these different classes of vaccines, so a titer is a measure of how much neutralizing antibody is produced. So a larger titer means that more neutralizing antibody has been produced. So for most vaccines tested so far in humans, the neutralizing titers have been similar to those tested with the same vaccines in small animals. They have been low for both inactivated virus and the adenoviral vectors, better for mRNA, and highest for the subunit vaccine. And higher neutralizing antibody titers are associated with higher protection, but it's not a linear relationship. And the reason that all the mutant strains, which you are all aware of, the so-called variants of concern, why they are a concern is those mutations affect the ability of neutralizing antibodies to bind to the spike protein of the virus. And the very recent Omicron strain has a very large number of mutations that result in escape from neutralizing antibodies generated by prior infection or vaccination. Of course, antibodies are not the only means of protection. And so even if, so therefore it's expected that vaccination and prior infection will offer good protection against the Omicron strain as well. This remains to be seen. And so virtually all the current formulations require refrigeration or freezing prior to administration. So these are the different variants of concern. Don't look at all the numbers, and they're not so relevant. In yellow are the mutations which occur in the part of the spike which binds the ACE2 receptor, that is called the receptor binding domain. So you can see that in the beta variant had 3 mutations in the receptor binding domain. The delta variant had 2. The Omicron has 15, out of which 10 are in parts which directly contact the ACE2 receptor. So you can see that clearly there is ongoing viral evolution. And with these mutations, there's a drop in the neutralization titer in convalescent sera as well as vaccinees relative to the corresponding titer against the wild type virus. And so in the Delta variant, there was a 2 to 7-fold drop, whereas in the Omicron, it's about a recent publication or preprint shows it's 10 to 15-fold higher than the Delta variant. However, we still don't know where, if mutations affect transmissibility, cellular immunity is also there. So we still do not know what the net result will effect of all these mutations will be in the real world on actual disease and hospitalization. So our candidate vaccine antigen when we started work on this in the beginning of February in 2020, we selected the receptor binding domain because it was very early days of the virus at that time. But going from our prior experience with flu and with HIV, we figured that antibodies directed against the receptor binding domain would be the most potent. And that has largely turned out to be true. And if you look at later on, after a lot of monoclonal antibodies have been found, in color are the regions on the receptor binding domain where one or the other neutralizing antibody binds. And basically the entire surface virtually of the receptor binding domain are targets for neutralizing antibodies. So not just the part that binds to the receptor, but many other parts of the RBD are also targets for neutralizing antibodies. So it's about 200 amino acids long compared to the whole spike, which is about 1,300 amino acids long. It contains 4 disulfides and 2 sites where sugar molecules can be added. And so we described early on in the pandemic, we made these glycan engineered fragments of the RBD. We expressed these in different kinds of expression systems and found to our surprise that the protein was very thermotolerant. So it wasn't thermostable, but when it was lyophilized, we could take the lyophilized formulations, the lyophilized protein, and heat it up even to 100 degrees. And when we cooled it down and redissolved it in buffer, it was just fine. So this was, potentially quite an important result, because unlike many of the other vaccine formulations, this meant that we would not require a cold chain. So we also found that while the monomeric protein did well in guinea pigs, it was not so immunogenic in mice. So then, using structure-guided design, we made trimeric versions where we fused a trimerization domain to one terminus of the RBD. We fused a number of different trimerization domains. This gave us one of the better results. We also expressed it on nanoparticles, because nanoparticles are believed to give better immunogenicity. And so what we found was that the protein formed very clean trimers, which were disulfide linked. So you can see the difference between reducing and oxidizing conditions. And other trimerization domains were more heterogeneous. And we also did animal studies with these molecules, and I'll just briefly summarize that work. Okay, so we first made the monomer, then we made the trimer, and we showed that the trimer was much more immunogenic in mice, and we were able to get neutralization of all 4 variants of concern. That time there was no Omicron variant. And so this was, and also protected hamsters from high dose challenge with replicative virus. Now, one drawback of the trimeric immunogen is that you also get antibodies against the tag sequence, which is used for the trimerization. So we wanted to avoid all kinds of extraneous antibodies, and we also wanted to see if a monomeric immunogen, which didn't have any trimerization domains, could we engineer it to make it more immunogenic. So I discussed briefly the saturation suppressor work we had done with CCDB. So we applied the same approach to the receptor binding domain. And again, we were able to make a long story short, we were able to isolate several individual stabilizing mutations. And when we combine these individual mutations, we got so these are different combinations of mutations. So we were able to increase the thermal stability by about 8 degrees or so. And all these mutations, it's a little difficult to see here, they're in red. They were away from the ACE2 binding site. So one mutation is close to the ACE2 binding site, but points in a different direction. So using yeast surface display, we were able to isolate a triple mutant, which was about 8 degrees more stable. And so this is just the distribution of the TMs of the single mutants and the TMs of the multi-mutants. So we hit a plateau at around 8 degrees. We couldn't really go higher than that. For reasons which I don't have time to explain here. But nevertheless, we then looked at the thermal tolerance of both the wild type and the multi-mutants. So the wild type, this is after 2 hours of incubation at different temperatures. We cool it down and look at the thermal unfolding. So as you can see, with more, as we incubate at higher temperatures, we get a loss of activity of the wild type. and so basically above 40 degrees, we start to see loss of activity. With the mutant at 50 degrees, which is 10 degrees higher, we still see no loss of activity with both of these mutants. So next is after incubating at 50 degrees for different times. So once again, you can see that with this stabilized mutant, even after 20 hours at 50 degrees in solution, there's almost no loss in the thermal stability. Whereas for the wild type, you can see there's a big drop in the amount of folded protein. So next, we looked in formulation with an adjuvant, and this is an oil in water emulsion called SWE. So what we were able to show is that our stabilized mutants, both in buffer as well as with the adjuvant, which is an immune stimulating molecule that we will be putting in the final vaccine formulation, stabilized it so that even at close to 2 weeks of incubation at 45 degrees, so this is 2 weeks incubation. The blue is at 4 degrees. This is at 45 degrees. We were still able to get quite a lot of retention of um, folded protein. And here what we are measuring is the ability of the protein to bind to ACE2 receptor. So this is the on rate and this is the off, the association and the dissociation. And so this plateau region is a measure of the amount of folded protein that is left after the incubation. So you can see compared to the wild type starting RBD, we are doing much better with the stabilized mutant. And this is without the addition of any other additive. Um, so again, what we showed, I will, in the interests of time, I'll just skip a lot of this. But essentially, we showed that we could express this protein, which I'll call MRBD1-3.2. This is expressed in mammalian cells and in Pichia. And what we showed is we could get very clean monomeric protein, which bound to a lot of conformation-specific antibodies. And both the mammalian cell expressed protein as well as the Pichia expressed protein were stabilized. And even after extended thermal stress at 37 degrees for a month, it retained both the mammalian and the Pichia protein retained binding to ACE2. So the reason we chose Pichia is that it's a much lower cost expression system than mammalian cells. Okay. Now, we next looked at the immunogenicity in mice. And again, I'll just stick to the main points. So this neutralization titer is the dilution of serum, which is able to prevent 50% of the virus from entering cells in cell culture. So for the wild type, you can see that the neutralization titer was around 100. This is a log scale. Whereas for our mutants, the neutralization titer was closer to 100,000. So we've gone up almost 100 to 1,000-fold higher in neutralization. The mammalian cell expressed protein had a bit better neutralization than the Pichia expressed protein, but even the Pichia expressed protein was pretty good. These are the neutralization titers now against all these different variants of concern. Right? This is the wild type, and this is the alpha, beta, and the delta variant of concern. And you can see that we got with our stabilized protein, it didn't matter which variant we were looking at, we got the same quite high neutralization titer. With the Pichia express stabilized protein, the titers were a little bit lower, but they were still very respectable, of the order of 10,000 or so for many of the variants. These are the values with replicative virus. Okay, so in red is for pseudovirus, in blue is for replicative virus, and even for replicative virus, we were around 1,000 or so. Whereas if you take now human convalescent sera, and these are high titer convalescent sera, which were provided by our collaborators, you can see that for the wild type virus, compared to the beta and the delta, the titers go down by quite a large amount, in contrast to the situation with our antigen. Of course, this is mouse sera and this is human sera. We can't compare them one to one. But nevertheless, these results are quite encouraging. We also did a challenge in collaboration with Dr. Amit Awasthi at THSTI and Dr. Zaigam Rizvi. And so these are now the body weight changes in the unvaccinated but challenged mice. And you see they rapidly go down and the mice die basically after 5 or 6 days. Whereas in the unimmunized and unchallenged mice, they gain a little bit of weight. And in our vaccinated animals, G1 and G2, those groups, they again are pretty much retaining the weight. If we look at the lung viral titers in the infected but unvaccinated animals, they are quite high as expected. And in our immunized animals, they are like the uninfected animals. So this was encouraging. We also did experiments in hamsters, and what we saw in the hamsters again was that there's a lot of data here. In the interest of time, I'll skip a lot of it, just to show that focus on the main things. These are the clinical signs. This is with the mammalian animals immunized with mammalian cell express protein, animals immunized with Pichia express protein. In the viral challenge control of unimmunized animals, there are very high clinical symptoms, whereas in our immunized animals, there are transient symptoms which disappear by day 4. The lung viral load is also log orders lower than the in the immunized animals compared to the unimmunized animals. If we look at the lung pathology, you can see that this is just different magnifications, but even to the untrained eye, you can see in the healthy animals, there are these large alveolar spaces, which are all gone in the unimmunized challenged animals, but they're retained in the vaccinated animals. This is for the mammalian cell express protein. This is for the Pichia express protein. So both the mammalian cell and the Pichia expressed antigens protect hamsters against high dose pathogenic challenge. So just to summarize, there have been a number of vaccines which have been granted approval in various geographies, and they all have good interim and in some cases confirmed efficacies, but mutations are a concern. It's already been seen that with the beta virus and more recently now with the Omicron variant, that there's a very large drop in neutralizing titers. And the current molecules that we have are highly immunogenic and thermotolerant. The neutralization titers we see in small animals are much higher than in convalescent sera. And the sera neutralize, again, we don't have data with the Omicron yet. We're trying to make all the necessary mutations. Hopefully in a few weeks, we should have some data. So the sera neutralize all the 4 variants of concern that were there when we were doing these experiments in both pseudoviral and replicative viral neutralization assays. We've made permanent cell lines, both mammalian cells as well as in Pichia, and such thermotolerant RBD derivatives show great promise to combat COVID-19 and likely do not require a cold chain. So we are now moving forward into clinical development, and this was considerably delayed because we had to raise the necessary funds to do this. It is not very easy for a small academic lab and a startup to do, but now we are on track. And we are in the process of developing further improved and more broadly protective coronavirus vaccines, because it's clear that this is not the first coronavirus to create a serious respiratory disease, nor is it going to be the last. So finally, I would like to acknowledge the many people who contributed. These are all people from my own lab at IISc, Samir did a lot of the initial monomer and trimer work. Dr. Rajmani did all the uh, BSL-3 challenge studies, except on the ACE2 mice, which were done by Vijayraghavan in THSTI. Shahbaz did the stabilization by yeast display with the help of Suhail, and a number of other people have contributed. Some of the structures, which I didn't show you, were done by Ishika Pramanik by cryo-EM in Somnath Datta's lab, and Shashank and a couple of his students contributed the virus, the challenge virus for the initial hamster, for the hamster studies. At THSTI, some of the initial neutralization assays were done by Shailendra Mani and Shankar, and the mouse challenge studies were done by Zaigam and Amit. At CSIR IMTech, all the pseudoviral assays have been done by Sahil and Rajesh Ringe. And the human samples, the convalescent sera, were provided by Rajesh Pandey and his collaborators at the Max Super Specialty Hospital in New Delhi. Some of the other viral neutralization assays were done with replicative virus in the lab of Dr. Vasan at CSIRO Australia. And a lot of the work was the guinea pig and mouse immunizations, protein purification, and serology were done by at Minvax, which is a startup I co-founded 3 years ago. These are the publications, our initial thermotolerant monomer, the later trimer, and the most recent stabilized monomer. And they've all achieved a fair amount of attention. And finally, I would like to thank for funding the Bill and Melinda Gates Foundation. There was a very crucial grant we got from the Office of the PSA, the Prime Minister's Scientific Advisor, and Minvax got a little bit of CSR money from Citibank, not Minvax, but SID at IISc, which enabled Minvax to do some of this work. So I'll stop now, and I'm happy to take questions. Thank you.
Thank you, Raghavan. Fantastic talk illustrating the transition from protein folding to a really exciting new vaccine. So I don't see anything in the chat box here. I suppose the questions might actually pop up from the YouTube. Can somebody from the check channel for any questions? If not, let me just start while I just get somebody to cross-check.
Yes, ma'am, I will do so.
There is no question in YouTube.
Thank you. Thank you. It's just that customarily these lectures which invited and given by fellows are not subject to too many questions, but it is such a hot topic and such an interesting topic that I want to take the time available. So Raghavan, a question on how much does the glycan contribute towards the protein stabilization, trimerization, and the immunogenicity, or in your recombinant vaccine, which is from Pichia, these glycan residues are not present?
Yeah, it's a good, very good question. So we, of course, also tried, one of the first things we tried was to express this in E. coli, and, we just could not get it to refold. And so the glycans are clearly very important for folding. Finally, in Pichia, what we were able to do was we made in our stabilized mutant, triple mutant, we could actually get rid of the glycans. And so there we found the stability drops by about 5 degrees or so when we remove the glycans. So it comes back to the wild type level. Now, if we look at the immunogenicity, the glycans in Pichia are a little bit different. They're high mannose glycans compared to the glycans in CHO or 293 mammalian cells. So what we found was that the immunogenicity of the mammalian cell produced protein was a bit better. Now, there were a few extra glycans in the mammalian cell protein. So we've now made the exact version, corresponding version in Pichia, and we are just in the process of getting the immunogenicity data. So at the moment, the Pichia molecules are a little bit less immunogenic than the mammalian cell expressed molecules, but they do have certainly several potential advantages. You can make a recombinant Pichia clone
Large
much
amount.
faster than a mammalian
Right.
one, and they can be grown to very large
Right.
volumes. So there are some advantages. So at the moment, the mammalian ones are a little bit better. We hope in due course with further, but the Pichia ones are also not bad at all.
With respect to the human trials, does it matter? Either one of them would work?
Well, so far
Or you
the
have to
small
apply
animal,
separately in the...
so far the small animal data has been somewhat predictive of human data. So to that extent, the mammalian one might be a little bit better, but one has to, balance all these things about, the ease of manufacture,
Correct.
the volumes and so on. So yeah, but at the moment we are going.
Did you do an NMR? Did you do an NMR of these proteins to know whether these glycans are present?
No, so we did mass spec, so we know the glycans are present.
Okay.
NMR has been, because you need labeled protein and it's not so easy to do the labeling in mammalian cells. So, yeah.
Great. So one last question, because I don't see anything from the YouTube channel.
Sure.
So are you again planning to do an exhaustive, all possible mutations in your RPD domain that you already have?
We've already done that. We've already done that, Usha. So the way we do it is we
so
make
any
single site
predictable mutant.
So we made all these mutations and then use the suppressor mutagenesis to isolate the stabilizing ones. So what I showed
No, I'm
was
talking about the variants, to all potential variants that could arise in virus evolution.
Right. See, one can do that. It's not clear to me that that's so worthwhile. There's a whole issue of gain of function research, because what's going to come up in nature is not necessarily, what you select
Yes.
in an experiment. So we have not
Yes.
bothered to do that, but
Okay.
it's something we could do. So.
Great. Thank you so much.
Sure.
And thanks for finishing well on time and engaging with my questions. I'm sure the participants and all of INSA joins in me in thanking you for the seminar and your time.
Okay, thank you.
Bye.
I'll try and unshare the screen. Bye. Yeah.
Yeah. So friends, we have 2 minutes before we are joined by the next fellow who is going to talk. So in case you need a quick break, we will start in 2 minutes. Hi, Vani.
Hi, Usha. I have.
You want to try and share your slides while you still have a little time?
Sorry, it's not. Oh. Just a moment. I think I didn't open the desktop. To the desktop. Usha, I have to sign out before, because I have to open a presentation on the desktop.
Okay. So you can sign out and sign back in right away.
Yeah.
Ma'am, you can open it without signing out.
Okay.
You can.
Yes, ma'am.
Should be possible because you open your PowerPoint presentation, go to share, and after that you select the
I've
application.
opened,
PowerPoint.
I need to, okay, here I think.
You can just minimize this WebEx window and then try to open your slide on the desktop.
How do I minimize this?
Hello.
Yeah. Just a minute. Got it.
Hello.
Yes. Yeah, I got it. Can you see my
Got
screen?
it? Okay. Yes, of course. So you can go to full screen mode.
Yeah.
Great. And in the bottom, you should be able to see the pointers somewhere. So usually it's in the bottom left. There will be a laser pointer option. Yeah, that's it.
Yeah.
Perfect. Okay, great. So are you ready? Shall we start, Vani?
Sure.
Short introduction to Professor Vani Brahmachari. Vani is a fellow of INSA, also of all the other academies of in India. And she's also served INSA in various capacities, including in the sectional committees. So Vani did her PhD at Indian Institute of Science, and her PhD research work was recognized by an INSA Young Scientist Medal. So she has been someone who has been on INSA's top list for quite many decades. After her PhD, she went for a postdoc at the Vistard Institute at Philadelphia, and then came back to IISc and joined IISc in 1990, the Unit of Developmental Biology and Genetics, and was there at DBGL, as it was called those days, till 1998, at which time she moved to Delhi, moved on to the Ambedkar Center for Biomedical Research at University of Delhi, championed and spearheaded the integrated master's PhD dual degree program. Through her work, both at IISc and ACBR, she has mentored so many PhD students, I think about 20 to 25 PhD students, guided and taught many master's students, and has done fantastic work in bringing high quality science to the next generation of India's scientists, the younger fellows. I now invite her to talk on a topic which is close to her research interests, that is on epigenetics and its intersection with development and disease. Vani?
Thank you, Usha, for that nice introduction, and also to give me this chance to share some of our research with the co-fellows. Now, our interest has been in the area of epigenetics, and I got introduced to this, in fact, at IISc way back in 82, 83, working with Professor Sharat Chandra in this on this fantastic system, the mealybugs. From there on, when I moved to Delhi, I started exploiting the genome sequence in order to mine for new regulators of epigenetic control. And what I'm going to talk today is sort of a concise picture of what we have done over these years. So these are the various areas we looked at with the main interest to consider the phenotype to genotype correlation, and also looked and focused mainly on chromatin modifying factors, and also in looking at in collaborative projects with epigenetics and complex diseases. What I'm going to talk about is this aspect of mining the genome or exploiting the genome sequence to look at the novel components of epigenetic regulation, primarily in the human genome, and try to validate this in suitable systems as in when we need to validate. Now, development is programmed and well studied in Drosophila, and the gene cascade, which starts off with maternal effect genes, comes down through the various genes to the homeotic gene expression, giving identity to the cells and the tissue. And one transition in the regulation that happens is that the initial genes have gene-specific regulators control them, whereas this transits to global regulators subsequently. Now, the global regulators, as one can conceive, can have, they generally work in complexes and therefore will afford, you can have a few of them coming in various combinations and bringing in a functional specificity. So our interest was to make use of the genome sequence in order to see whether we can identify new members of these complexes. And when we started around the 99-2000, there were very few such genes, especially the regulatory complexes going into what constitutes the developmental regulatory complexes, polycomb and trithyrax, in the database. Now, one of the confounding factors in this kind of search is that you cannot not always apply a homology search-based approach for identifying these, because the functional class assignment can be very different from the biochemical function. What I mean is something like when we talk about oncogenes and tumor suppressor genes, this is the cellular effect or the end effect of these genes, and they are classified under these categories, whereas the biochemical activity of these genes is itself very different. Similar thing happens with the polycomb and trithyrax genes as well. And as you see here, this was, I mean, it's too familiar with audience. Tumor suppressors can have various kinds of biochemical activity, so also the oncogenes. Now, that time, the trithyrax gene similarly, this group had lots of members, but then their activity, biochemical activity, differed from each other. So the typical homology search might not yield the information about, though they will yield about their biological activity. However, it's hard to get the information on their cellular activity. So we took an approach, which is a peptide identity search, which was through a software called the PL host, designed at the IGIB. So basically, we made a pool of all the peptides coming from the various known trithyrax and polycomb members, and then look for identity signature of peptides which are at least 8 and more, so that the random probability occurrence decreases. And then use this sequence to look at the genome, identify that, use that as a signature, and identify the proteins that contain this signature. So when we did this with the polycomb and trithyrax proteins known at that time, we identified a 14-mer peptide, which was there in many genomes, starting from the yeast to the humans. And among them, there were some which were already assigned with, annotated, and there were others in each of these organisms which were not annotated yet. Once a gene was the gene on the chromosome 15, which was identified as a homologue of the INO80, it was known in the yeast system. Now, the peculiarity of this was that our in silico analysis showed that apart from the peculiarity in the spacing that was already known from the yeast studies between the various domains, we also identified a potentially DNA binding region, which we called as the dipinology. And when we looked at the phylogeny of this, we could see that this forms, the INO80 forms a distinct subfamily within the SNF2 chromatin remodeling complexes. So this is figured in the NCBI database. So basically, our effort was to continue and validate the predictions that we made, namely that this is a DNA-dependent ATPase, and it shows direct interaction with DNA, and also look at the cellular localization of this protein. Now, the major interest we had was whether this protein participates and controls development. And to this, we have to go back to the, the wonderful system of Drosophila, which we utilized with the collaborators. And we found that the DNO80, the Drosophila homologue of this, is expressed everywhere, and it localizes to many sites on polythene chromosomes, suggesting that it interacts with the chromatin. And null mutants of DNO80 were late embryonic lethal, and we also detected the misexpression of homeotic genes. In the early stages, what was interesting was that in early development, the INO80 acts as an activator protein, but for the same gene in the later stage, we found that it acts as a repressive protein. And we also found that the INO80 interacts with both the polycomb, which is the repressive complex, and the trithorax proteins. And these proteins have been given a separate class, which is called the enhancer of polycomb and trithorax. Now, this is the result I was talking about, in which you see that this protein sex comb reduced, it's a meiotic gene, in which you see that the wing disc shows it acts like an activator, whereas it acts as a repressor in the other imaginal discs. Thus, it seems to have a dual function, and the way it achieves perhaps should be through the polycomb and trithorax complex interaction. So then the interactions are also well easily studied in Drosophila, but the complexes that mediate such repressive and activating effects on a large number of genes is referred to as the cellular memory modules. These basically maintain the gene activity state through development. So there are transient signals to which the cell responds. To sustain this transient change in the expression, cellular memory modules come into play. And in development, all the more we can see the importance of this, because the early determination of the cell development, or the determination step, has to be retained through the development, even though the initial signal disappears. So this is a typical repressive complex, which is called the polycomb repressive complex, and this is the activating complex, which for reasons of the phenotype one sees in Drosophila is called the trithorax complex. So the sequences to which these bind are referred to as the polycomb responsive element or the trithorax responsive elements. And interestingly, one finds that there are similar complexes, protein complexes, which have been identified in the human as well. So ultimately, the maintenance or the sustenance of the activity state is brought about by histone modification, and the mediation of in these complexes, generally there is a catalytic activity of histone modification, either acetylation or methylation, that is associated. You also have several complexes. This is the activating complex or the trithorax complex. As one can see, you have Drosophila and the human complex both have very similar kind of components, and the modification they bring about is the K4 methylation on histone H3, which is an activating signal. So the human genome also codes for many of the components of this activating complex, which is similar to the trithorax complex. Now, the building blocks for, if you generalize the composition of a complex, either a repressive complex or an activating complex, one would see that you need, one of the components needed is a DNA-dependent ATPase, because the energy is used to open up the chromatin or condense it. And you also have the catalytic activity, histone or DNA modifier. And there are several actin-like proteins which are part of it. And you have a critical component which can interact with DNA, which one can call as a recruiter, which brings to the site of action. Now, this can be generalized both for an activating complex as well as a repressive complex. And the recognition sequence to which the recruiter brings it, and generally it is not a single recruiter and multiple sequences are involved to reinforce the system. And these sequences are the ones which I refer to as the PRE or the TR, responsive elements. So this is the common sort of terms that one can think of, and therefore the DNA-dependent ATPase is required both in the case of repressive complex or an active complex. So in order to look at the functional diversity of D-anoity, the Drosophila homolog, we studied the genetic interaction in of anoity in the Drosophila system, and the target genes can switch to an inactive state or an active state depending on what is the interaction that is mediated. And there are these fence that are sort of either interact with the polycomb or the trithorax complexes. Now, this was a series of an extensive analysis of genetic interaction that Morrison carried out. And basically, your readout is that of the morphological defects that you see in the abdominal fusion or the wing morphology or the sex chromes on the leg. So in this analysis, the outcome of this analysis was that you can see that the anoity interacts with both the repressive complex as well as the TRX, that is the activating complex. And there are other enhancers of the trithorax and the polycomb protein, which are also, and anoity can be considered as a member of this. Now, one of the caveats of this is that these are genetic interaction. So there can be, it doesn't imply that there is a direct interaction between anoity and all these proteins. But however, it is reasonable to assume that such an interaction probably occurs. Now, at that time, what was on our mind was what is the DNA binding domain of anoity doing in recruitment? Does it have a role and so on? So, but what was known in literature was this highly ubiquitous transcription regulator, yin-yang-1. Its homolog is called a 4 in Drosophila. This is known to be a sequence-specific DNA binding protein and is part of the anoity complex. Now, question we were interested was to address whether the function of the DNA is always dependent on 4. So if you take the null, if you consider the lethality that occurs in 4, the 4 mutants are pupil lethal. At the pupil stage, they die. Whereas anoity we found through the p element excisions that it is early development, late embryonic stage lethal that we find. So this has got its place. We looked at both the knockouts as well as the knockdowns for both of these genes. Now, when we looked at the what are the kinds of changes interacting networks that these kind of mutations bring about, especially the INOAT that we studied, we found that if this is the wild type network that we see for the selected proteins that we looked at, including the homeotic genes and the regulators of homeotic genes and so on, we find that this is a kind of network that you derive, while the whole structure drastically changes when you deal with the INOAT null mutants. And as we could see, there are certain polycomb proteins also in the network whose interaction are altered, and as well as the epigenetic modifiers like the histone deacetylase or the histone methylases. Similarly, at a later time point, because this is a late embryonic lethal, you also see drastic change in the protein networks. Now, this correlated with some of the phenotypes that in some cases, earlier points, we could show that the correlation between the phenotype and the loss of this interaction. Now, with reference to the DNA binding domain and to look at its interaction with DNA and so on, we concentrated on what we called as the DBNO domain in various kinds of experimental context. So the hypothesis that we wanted to test was whether you can have, there can be complexes that have HINOAT where it is doing only its chromatin remodeling function and gets recruited by perhaps different proteins and also the YY1. And it can be the same complex wherein it can also be targeted to regions where there is a HINOAT binding site as well as a YY1 site, and the complex can get recruited at that point. And it's possible that the composition of the complex also varies. So basically, it brings about, with a few proteins, it brings about a variety of interactions and a consequence of that, variety of regulatory activities in the system. Now, we could demonstrate through the typical EMSA assays and so on that the HINOAT, the homologue of and its homologue in the Drosophila, directly interact with DNA through a consensus sequence, as shown here. And they also tolerate certain changes at some of these positions, which we could demonstrate. And in a cellular system, they can alter the reporter gene expression when it's cloned upstream of the promoter. So in other words, the DNA binding activity at the HINOAT in its own right recognizes a sequence and binds at that position. And whether this happens as a sort of a moonlighting additional activity of INOAT, or it is forming complexes of these kind where YY1 is not involved at all, is a question that we are currently addressing, basically through a serial sort of IP exercise where you remove all the complexes containing YY1 through using the antibody against YY1, and then try to pull out complexes which are containing the INOAT. So this is ongoing, and one of my students continues on this line in his independent position. Now the INOAT regulates homeotic genes, so it was important to see whether the homeotic gene regulation through INOAT is also mediated by the PRE and TRE sequences. And in this effort, again, we resorted to looking at the Drosophila system and studying the transgenic fly, which has the IAB7 PRE upstream of the eye color gene, and to deal with mutants of INO80 and see whether it brings about any effect on the eye pigmentation. As you can see in the cases, example cases given here, there is an effect, or repressive effect is relieved when you bring in a mutation in the immunity. And this effect is seen when you see the bring it along with the mutations FOO and ASX. So, in other words, what we could derive from this is that the PRE, that INO80 interacts with the PRE, TRE sequences. So this, having seen this effect, now we can see that the INO80 can function as a silencing or an activating factor, and therefore it has a dual function in that sense, as well as it has the activity of DNA-dependent ATPase and functions like recruiter in certain cases. This needs a further substantiation to show that this is happening in absence of any other recruiter. Therefore, so far what we have done is identify the human homologue of INO80 and shown that it confers developmental memory as a part of the polycomb and trithorax complex, and also a direct interaction of this protein with DNA promises that there can be multiple functions that INO80 does, whether it is like a moonlighting function, which is not a very usually done, but only under certain circumstances. Such an activity is also known for another member of the trithorax family, that is the MLN. Now, this part of the work was carried out by these young students during their PhD, and some of them as postdoc. Rachit started this project, and Shipra, Abhishek, and Shruti continued. Mohsen has shown a lot of the fly studies, and Shweta has worked on the binding of INO80 to DNA and immuno function in the mammalian cells. We had a very fruitful collaboration with Professor Shanti and also with Rakesh Mishra in his group for Vasanti and Narendra. Now, there is another part to this whole regulation that is important. That is the role of the cis element, which is the PRE-TRE. Now, it's an important counterpart of epigenetic regulation. And its function is, this sort of brings in the paradigm of the operator and the promoter overlap that you see way back in the lambda phase. So here is one sequence which recruits primarily the repressive complexes, and this is the one which recruits the activating complexes. But when you think of it, it is basically a DNA interaction, and then one does not see readily how the binding of these proteins, the specific single protein, would bring about these two differential states. But it is a complex which probably is influenced. When we started looking for these things, there were not many. There were 2 reports of the PRE-TRE in the human case. And we wanted to look at this and see whether we can identify new members of candidate sequences which can function like the polycomb responsive element or the trithorax element. Now, what is the strategy we applied for this? One of them was to use a program which was basically designed with Drosophila as the sequences as the training set. And Other one was to look for the density, high density of occurrence of the Y1 binding site. And yet another, by the time the ENCODE data came, one could look at the distribution of epigenetic marks and also look at the chip data wherein you see the localization of the polyclonal protein or MLL and so on, and putative PREs and TREs could be derived or could be detected through such a site. Now, the data set for the search, we could have used the whole genome, but we went for a selected region search, especially with reference to using this approach of predictor as well as searching for binding sites for this specimen. In fact, for all the three cases. Now, what we applied, the logic that we applied here was that to use those genes which are misexpressed in leukemia patients with the translocation 411. Now, why this particular translocation? Because chromosome 11 and the region that is involved in this translocation contains a gene which is very similar to the trithorax gene. That is the MLA. It's an activating protein. And therefore, we thought that the genes which are misexpressed due to these translocation may bear the upstream sequence which can interact with activating complex. So with that logic, we looked at both the downregulated as well as the upregulated genes in these patients as against the normal individual, normal cells, and tried to identify the potential sequences. What was interesting was we also looked at the YY1 binding sites within these regions, and we could identify, especially with reference to PIK3CB, similar regions by both approaches. And we concentrated on this. Akanksha, who worked on the third kind of approach subsequently, concentrated on homeotic genes and identified putative PRE in HOTSP3, a big complex. So in this attempt, what we did was for the PIK3C2B case, we looked at the density of these motifs and then picked up one of the highest density was in this gene, PIK3C2B. And this particular sequence was interesting because it had about 25 repeats. Yeah. So we had the binding site repeated 25 times along with a site for the GAGA protein binding. So, which is the activating complex member. So this was the binding site for YY1, which could bring in a repressive complex, especially the PRC2, which can bring about methylation. And this one, that is the GAGA binding protein homolog of the mammalian system, which had been. So this whole sequence was within 1 kb was repeated 25 times. So we chose this for further validation as well as the usage to check whether we have identified a putative PLETI. PIK3C2B, not much is known about the gene itself, but then it is influencing cell proliferation, survival, migration, and so on. Now, we went through the validations and demonstrated the regulatory function of this sequence, which we call the PRE PIK3C2B, in transcription and in the recruitment of PRC2 complex. And the repression is correlated with the histone methylation, that is the trimethylation of K27 on H3. And in transgenic Drosophila, it functions as a typical PRE and also one of the rare mammalian PREs, which brings about pairing sensitive silence. So one could see that this was the transgenic work that was done in collaboration with Rakesh. And we could see that it functions as a PRE. So our confidence to call it a PRE came from the experiments in the Drosophila system. Now it interacts, genetic interaction with many members of the polyhuman trithorax family, we could demonstrate. And also it has both a repressive and an activating complex interaction, and it interacts with activating complex proteins even in the human cells. Now this was analyzed by mass spec, the proteins which interact with these sequences, and through an approach of using 3 different cell lines and repeats, the common ones were common proteins that came up were looked at. And it was interesting to see many of the histone modifiers as well as other proteins, the ARPs, coming in this analysis. Now, this is being taken further by Jayant, who started the project lab. So what was interesting was that this also responded, the sequence PIK3C2B responded to the activating and repressive complex more or less like the, what we would see in the lambda system. In other words, when you knock down one of them, right, then you can clearly see that the occupancy of this site by, let's say, if you knock down the trithorax or the activating complex, then the occupancy of the PRC proteins or the repressor proteins becomes higher. And similarly, when you knock down the repressive complexes, you have the members of the activating complex occupy these sites, thus demonstrating that this can switch from activate state to a repressive state and responds to the cellular concentrations of the respective proteins. So it shows a dual regulatory function, and it also has the correlated histone marks like H3K4 trimethylation for activation and K27 trimethylation for repression. And similar interactions as well as activity was seen with reference to Drosophila. And once again, it showed the interaction with PCG proteins, the trithorax proteins, as well as the ETP. So therefore, there was very much of a similarity in the way this particular sequence behaved, both in case of Drosophila, when it was in the background of Drosophila, as well as human cells. And the next question was, how far does it, for this sequence, exert its regulatory effect? The first step we did was to look at the 2 neighboring genes, one which was about 11 kb away, another which was 21 kb away. And we could see that these 3 gene expressions was coordinated, and they also responded to knockdown of YY1, which is the major recruiter, and they have the binding site for this. And therefore, that prompted us to ask whether it has long-range interactions. So I should be done soon. But the long-range interactions were examined in HEK cells, basically, and well, we could detect several intrachromosomal interactions. And more or less, the characteristics of these interactor sequences were analyzed by using the ENCODE data in 3 different cell lines, so that we want to see whether there is any consensus activity or correlation with regulation that we could see. In most of these cases, the what one would see is the heterochromatic nature or the low transcription was one of the features that these interactor sequences exhibit. Apart from the intrachromosomal interaction, we also detected the interchromosomal interaction, once again with the similar character of having much more of heterochromatic, low expressing, low transcription regions associated with the PI. So this to us suggests that whether it is making a hub of the heterochromatic regions, in this case, though it is capable of activation, and this data is getting confirmed by the 3C methods, that is validated, getting validated by 3C. And interestingly, we see that the interaction as 3C reflects that the orientation of the sequences with reference to the bait, namely the PRE-PIK3C2P, is really varied in each case. In a given case, you can see it that their topology of interaction varies. This comes from the data which shows with the PCR primers mapping at different position between the bait and the targets. So basically, this is where we are with reference to the identification of the PRE sequences. And Akanksha has some interesting results with her PRE. I have not covered her work in this. And the project got started by Hemant, and Jayant has carried out a lot of the mass spec as well as the 4C experiments and analysis. And Mosam was the fly person in this group, and Kaushik did the data analysis. We had collaboration for proteomics with Suman Thakur, and where he sent us the raw data and we could analyze it. And Rakesh helped us with the transfer. So I would stop here. And what I'm glad is that some of these branches which show a promising sign of future is being followed, and some await confirmation for the work on that. Thank you very much.
Thank you, Vani, for a very, very illuminating talk, taking us from how to search and find chromatin regulators to actually showing functions and large chromosomal effects of these regulators. So I will just look into the chat box to see a couple of questions which have popped up. And then finally, I will go to one or two small questions that I have. There's a question from Professor Arunlairi Majumdar that do you plan to raise these antibodies through plant expression? So I think he's referring to INO80 antibodies or others.
Okay. Commercially, there is INO80 antibody for Drosophila, as well as the Drosophila is only with one of the investigators so far. But the human antibody is made by Vogen as well as another one. And we have found some cross-reactivity of the human antibody against the Drosophila protein. I haven't particularly looked at the plant systems as such, so I can't comment.
So while we will check if there are any questions in the YouTube channel, Vani, I was very intrigued by your very early part of the talk where you showed that there are sequences which are probably proximal to each other or in the same chromosomal region, one of which could bind YY1 and another one which could be recognized by INO80. Do you think that target genes which have both these sides show cooperative effects? Because there could be two different types of complexes binding there?
No, the distance, I don't know whether topographically, whether it would be possible for the two complexes to bind. But what we have seen in chip experiments is, I mean, they are not as close as I showed in the model as such, right? So we have looked at the Hox genes and their distances are more than 1,000. Some cases, but they are within 1,000 in other cases. And in these instances, we have sort of knocked down one of the proteins, tried to see whether there is any other complex binding there, or NOG itself can bind, which indicates that it can bind in absence of Y11. And we do see such cases. So in other words, these two proteins are not dependent on each other for the binding. That has been experimentally demonstrated. But we don't have evidence to show that both the complexes can occupy that region simultaneously. And of course, it remains to be seen because one is member of, both of them can be member of the same complex. It's
complex or different complexes. Yeah.
Both possible, but then it has to be dissected whether it is part of the same complex or both of them are being the same. But we do have cases wherein there are more than one DNA-dependent ATPase in the same complex, for instance.
Interesting. So another small question that came to me on the second part of your talk on the PIC2B. So in normal cells, the implication is that it is associated with regions that are more heterochromatin-like than expressed, and in cancer tissues, it switches to an expressed form. Is that correct?
See, we are depending to make these kind of assessments mainly on ENCODE data, right? So what we have is data on cell lines which have been maintained in vitro for many, many generations. So unless we do with primary cells, I don't think we'll be able to make that distinction as such. But what we see right now is that whatever the interactors that we see, mostly they are enriched in what I say, the repressive kind of marks.
Well, I don't see any other questions. In any case, this has been a talk which is an invited talk. We don't entertain too many questions. So it's been very nice to hear your contributions in probing, epigenetics in normal development and disease, and using wonderful collaborations and model systems ranging from flies to human data. Thank you, Vani. Thank you for sparing the time from both myself as well as INSA.
Thank you. Thank you.
And thank you all participants for joining this session.